cellular respiration depends on the flow of electrons to molecular oxygen. In the absence of such inhibitors, the flow of electrons is coupled to the transport of protons out of the matrix. The removal of protons from the mitochondrial matrix does two things. It creates a concentration gradient across the inner membrane as the concentration of protons outside the matrix exceeds the concentration inside. It also builds up an electrical potential because a positive charge is removed every time a proton is transferred out of the matrix. The combination of proton concentration and electrical gradients, called an electrochemical gradient, creates the proton motive force. The proton motive force is the energy stored in a proton gradient. When this energy is released, ATP is synthesized. How does the proton motive force make ATP? As you will see, there's another protein complex, complex 5, also known as ATP synthase. ATP synthase is a rotating molecular motor that contains a proton channel. When protons flow through this channel, ATP is synthesized. ATP synthase is made up of two distinct components. One component is in the membrane. It has a ring of alpha helices through which the protons flow. The other component contains the enzyme's 3 active sites. Protons flowing through the first component drive conformational changes in the second component. ADP and phosphate bind to one of the active sites. Conformational changes drive the ADP and phosphate together to form ATP and then force it out of the active site. In 1997, the scientists Paul Boyer and John Walker were awarded the Nobel Prize for their work on how the ATP synthase enzyme works. The newly formed ATP is transported out of the mitochondrial matrix. Its export is coupled to the import of ADP. ATP carries a greater negative charge than ADP, so its export is favored by the membrane's electrical potential. Like ADP, phosphate must also be transported into the matrix. The phosphate carries a negative charge. Would you expect the transport of phosphate into the mitochondrial matrix to be spontaneous? Select your answer and click Submit. Yes, that's correct. No, that's incorrect. Because phosphate carries a negative charge, its transport into the matrix is not spontaneous. Its movement into the matrix is coupled to the return of a proton. Now that we've seen how the proton motive force is used to synthesize ATP, let's learn how some of these details were discovered. When scientists discovered that organisms oxidized food for energy, they wondered how the oxidation energy was captured. Researchers eventually determined that ATP and electron transport were involved. Scientists proposed various models to explain how these two processes were linked. In 1961, the biochemist Peter Mitchell proposed the chemiosmotic model, which is the model we have just presented. Mitchell proposed that ATP synthesis is coupled to electron transport by the formation of a proton gradient. Experimental results support the chemiosmotic model, and Mitchell was awarded a Nobel Prize for his work in 1978. Let's see how the chemiosmotic model is supported by experiments that disrupt the proton gradient. Inhibitors are valuable research tools. These molecules are often used to study enzymes and have important applications in the development of new drugs. One class of inhibitors disrupts the coupling of electron transport to ATP synthesis. They're called uncouplers. Unlike inhibitors that bind to specific enzymes, uncouplers are hydrophobic molecules that carry protons across the inner mitochondrial membrane. Uncouplers allow protons to bypass ATP synthase and re-enter the mitochondrial matrix. The energy stored in the proton gradient is lost as heat rather than used to synthesize ATP. Uncouplers destroy the proton gradient and prevent ATP synthesis. Would you expect an uncoupler to affect electron transport? Click in the box next to the correct answer, then click Submit. Yes, that's correct. No, that's incorrect. Electron transfer reactions are spontaneous. The released energy is used to pump protons out of the matrix. The ability of the protons to flow back into the matrix without entering complex 5 has no effect on the transfer reactions. ATP synthesis is not spontaneous. It requires the energy released as the protons flow back into the matrix. Some uncouplers are made by an organism to regulate its metabolism. Uncouplers create a membrane channel through which protons can flow. Newborn babies have high levels of the uncoupler thermogenin to help them keep warm. Leptin is a small protein that helps regulate body weight. It's secreted by fat cells. Some people and animals that are obese have abnormal levels of leptin in their blood, or defective leptin receptors on their cells. In healthy animals, leptin levels rise when food calories exceed their needs. This does two things. Appetite is suppressed, and the levels of an uncoupler increase. The uncoupler speeds up the organism's metabolism, and more calories are lost as heat.